Common questions on the respiration topic in A-level biology AQA. So put a tick in the box next to the process that occurs in anaerobic respiration but does not occur in aerobic respiration. So reduction of pyruvate. A student used the apparatus shown in the diagram below to measure the rate of aerobic respiration of seeds for 48 hours. So you've got your respiring seeds here, wire gauze to hold the seeds, coloured liquid and potassium hydroxide to absorb the carbon dioxide. During the 48 hours, the coloured liquid moved to the left. Explain why. It's because oxygen is taken up by the seeds, but the carbon dioxide that's given out is absorbed by the potassium hydroxide. Therefore, there is a decrease in pressure inside. Apart from time, give two measurements the students would have to make to determine the rate of aerobic respiration of these seeds in centimetres cubed per hour. So, the centimetres cubed bit, they're going to have to find the distance that the liquid moves, but also the diameter or radius of the tubing. So from that, they can find the volume. Describe the process of glycolysis. Phosphorylation of glucose using ATP. There's then oxidation of triose phosphate into pyruvate. A net gain of ATP. So it would be a net gain of 2 because 4 are produced but 2 are used. And NAD is reduced, or you form NADH. Malonate inhibits a reaction in the Krebs cycle. Explain why malonate would decrease the uptake of oxygen in a respiring cell. Less or no reduced NAD or coenzymes, or fewer or no hydrogens, or the electrons are removed and passed to the electron transfer chain. Oxygen is the final electron acceptor, or the terminal electron acceptor. Yeast cells can respire aerobically or anaerobically. A student used the apparatus shown in figure 1 to measure the rate of respiration in yeast. She positioned the flask in a water bath so that the yeast culture reached a constant temperature, then left the apparatus for 1 hour before starting her investigation. Suggest one reason why it was important that the student left the apparatus for 1 hour after the yeast culture reached a constant temperature. So you can see here you've got the scale with the capillary tubing and coloured liquid, the yeast culture in glucose, but then they've got alkaline pyrogalol to absorb the oxygen. So it's so that all the oxygen is absorbed. During her investigation, the coloured liquid moved to the right. Explain why it moved to the right. It's anaerobic respiration, obviously because the oxygen has been absorbed, so there is no oxygen. Anaerobic respiration produces carbon dioxide. That increases the pressure or volume of the gas, which causes it to move to the right. The diagram shows a mitochondrion. Name the parts labelled X and Y. So X is cristae, part of the inner membrane, and Y is the matrix. During respiration, where exactly in a cell does each of the following occur? Glycolysis. That happens in the cytoplasm. The electron transfer chain happens in the inner mitochondrial membrane or cristae. Without oxygen, less ATP is produced by respiration. Explain why. Because oxygen is a terminal electron acceptor, So there's no electron transport chain or no oxidative phosphorylation, which is where most of the ATP is produced. In anaerobic, only glycolysis takes place, and pyruvate is used to make lactate, and there's only net gain of 2 ATP produced. The boxes in the diagram represent substances in glycolysis, the link reaction, and the Krebs cycle. Complete this diagram to show the number of carbon atoms present in one molecule of each compound. So it starts off with a 6-carbon compound, then 2 molecules of a 3-carbon compound, and then you get 2 molecules of pyruvate at the end of glycolysis. In the link reaction, you get a 2-carbon compound, and that 2-carbon compound combines with a 4-carbon compound to make a 6-carbon compound. It loses a carbon dioxide in decarboxylation to form a 5-carbon compound. Other substances are produced in the Krebs cycle in addition to the carbon compounds shown in the diagram. Name 3 of these other products. So you've got reduced NAD, reduced FAD, and ATP. The diagram shows the structure of a mitochondrion. In which part of the mitochondrion does the Krebs cycle take place? In the matrix. Name 2 substances for which there would be a net movement into the mitochondria. So either pyruvate, ADP, inorganic phosphate, reduced NAD, or oxygen. The mitochondria in muscles contains many cristae. Explain the advantage of this. Larger surface area for electron carrier system, oxidative phosphorylation, provide ATP or energy for contraction. Name substances X, Y and Z. So we're looking, X is here, Y is here, and Z is here. So X is going to be carbon dioxide because the pyruvate gets decarboxylated to form a 2-carbon compound acetyl-CoA. And Z is water. So oxygen and water is added to form water, added to the H plus and the E minuses. Also carbon dioxide X is given off here, decarboxylations turn it from a 6 to a 5 and then a 5 to a 4. So X is carbon dioxide, Y is 2-carbon acetyl-CoA, and then Z is water. Give the location of each of the following in a liver cell. Glycolysis happens in the cytoplasm. Krebs cycle happens in the mitochondria. Write the letter A on the diagram to show one step where ATP is used. So ATP is used right at the beginning of glycolysis to turn glucose into triose phosphate. Write letter B on the diagram of 2 steps where ATP is produced. So ATP is produced the second part of glycolysis. 4 ATP are produced, but obviously 2 are used as net gain of 2. ATP is also produced in the Krebs cycle, and ATP is also produced in oxidative phosphorylation. Apart from respiration, give 3 uses of ATP in a liver cell: as a source of energy or a source of phosphate, active transport, phagocytosis or endo or exocytosis, cell division or mitosis, synthesis of glycogen, protein or enzymes, DNA or RNA, lipid or cholesterol or urea. The main stages in anaerobic respiration yeast are shown in the diagram. Name process X, that is glycolysis. Give one piece of evidence from the diagram which suggests that the conversion of pyruvate to ethanol involves reduction. So here. It is because oxygen is removed from the pyruvate, reduced NAD is oxidised, or it donates a hydrogen where it donates an electron. Explain why converting pyruvate to ethanol is important in allowing the continued production of ATP in anaerobic respiration. It allows NAD to be recycled or reformed so that glycolysis can proceed, or so that more glucose can be converted to pyruvate so that process X can continue. Give 2 ways in which anaerobic respiration of glucose in yeast is similar to anaerobic respiration of glucose in a muscle cell. ATP is either formed or used, pyruvate is formed or reduced, NAD is reduced, and glycolysis is involved, or it's a 2-stage process. The flowchart shows the main stages in aerobic respiration. Complete the flowchart by writing in the appropriate boxes the number of carbon atoms in substance P and the name of substance Q. So we're looking here and here. So first of all, glucose is turned into triose phosphate, and then what is it turned into? So it's then turned into pyruvate, which is a 3-carbon compound. And then in the link reaction, because it's decarboxylated, it turns into the 2-carbon acetyl-CoA. Actually, you'll have to say the full version acetyl-coenzyme A, because AQA at the moment are very against using abbreviations. So acetyl-coenzyme A. The figure below shows the apparatus used for measuring the rate of oxygen consumption in aerobic respiration by seeds. For the first 10 minutes, the tap attached to tube A was left open and the syringe from tube B was removed. Suggest 3 reasons why the apparatus was left for 10 minutes. It's so that an equilibrium is reached, or to allow it to equilibrate, to allow for pressure changes in the apparatus, allow for respiration rate of seeds to stabilise. After 10 minutes, the tap attached to tube A was closed and the syringe was attached to tube B. Every minute, the syringe plunger was moved until the levels in the U-tube were the same. The reading on the syringe volume scale was then recorded. The results are shown in the table below. During the experiment, the coloured liquid in the tubing moved towards tube B. Explain what caused this. So it's moving this way towards the seeds. So oxygen is taken up by the seeds, but any carbon dioxide that is given out is absorbed by the potassium hydroxide. So oxygen's used, but any carbon dioxide that was produced is absorbed. So therefore the pressure is going to decrease in B, which causes the liquid to move towards B. The diagram represents 2 of the stages of aerobic respiration that take place in a mitochondrion. Name substance X. So it's something that is going in. You've got coenzyme A here going to acetyl-CoA. Something is then turned to, something is oxidised to form reduced NAD, and carbon dioxide is given off. So it's going to be pyruvate because this is the link reaction. So we want the product of glycolysis, which will be pyruvate. Which stage of aerobic respiration takes place inside a mitochondrion and is not represented on the diagram? It is the Krebs cycle, because here we've got the link reaction. Here we're talking about oxidative phosphorylation. Explain why oxygen is needed for the production of ATP on the cristae of the mitochondrion. ATP is formed as electrons pass along the electron transport chain. Or oxygen is a terminal electron acceptor, or accepts electrons from the electron transport chain. Electrons cannot be passed along the electron transport chain if there's no oxygen to accept them. It forms H2O or accepts H+ from reduced NAD. Describe the part played by the inner membrane of a mitochondrion in producing ATP. Electrons are transferred down the electron transport chain. This provides energy to take protons or H+ into the space between the membranes. Protons or H+ pass back through the membrane into the matrix through ATPase, and the energy is used to combine ADP and phosphate to produce ATP. Describe how ATP is made in the mitochondria. Substrate level phosphorylation. ATP is produced in the Krebs cycle. The Krebs cycle or link reaction produces reduced coenzymes like reduced NAD and reduced FAD. Electrons are released from the reduced coenzymes. These electrons pass along the carriers through the electron transport chain through a series of redox reactions. Energy is released, and ADP combines with PI to form ATP, and protons will move into the intermembrane space via ATP synthase. Table contains statements about 3 stages of respiration. Complete the table with a tick if the statement in the first column is true for each stage of respiration in an animal. So in the mitochondria, both the link reaction and the Krebs cycle occur. Carbon dioxide is produced in the link reaction and the Krebs cycle, and NAD is reduced in all 3, glycolysis, link reaction and the Krebs cycle. Some plant seeds can respire aerobically and anaerobically. A summary of the process of anaerobic respiration is shown below. Where in a cell does stage 1 occur? So glucose going to pyruvate is going to be in the cytoplasm because it's glycolysis. Explain how stage 2 enables stage 1 to continue. So it regenerates the NAD or oxidizes the reduced NAD to produce NAD. And NAD is reduced in stage 1 in glycolysis. A student investigated the rate of anaerobic respiration in yeast. She put 5 grams of yeast into a glucose solution and placed this mixture in the apparatus shown in the figure below. She then recorded the total volume of gas collected after 10 minutes for 1 hour. Explain why a layer of oil is required in this investigation. Prevents oxygen being taken up or oxygen being absorbed because we want it to be anaerobic. Describe how acetyl coenzyme A is formed in the link reaction. There is oxidation of pyruvate, or the hydrogen is removed from pyruvate, and carbon dioxide gets released. And then there's the addition of the coenzyme A.